Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Cell biology. 174 languages. Cell. Onion, allium sapar, root cells in different phases of the cell cycle, drawn by E.B. Wilson, 1900. A eukaryotic cell, left, and prokaryotic cell, right. Identifiers. Mesh D002477. THH1.00.01.0000001. FMA 686465. Anatomical terminology. Edit on Wikidata. This article is about the basic unit of life forms. For the branch of biology that studies them, see cell biology. The cell is the basic structural and functional unit of all forms of life. Every cell consists of cytoplasm. Enclosed within a membrane, many cells contain organelles, each with a specific function. The term comes from the Latin word cellula, meaning small room. Most cells are only visible under a microscope. Cells emerged on Earth about 4 billion years ago. All cells are capable of replication, protein synthesis, and motility. Cells are broadly categorized into two types: eukaryotic cells, which possess a nucleus, and prokaryotic cells, which lack a nucleus but have a nucleated region. Prokaryotes are single-celled organisms such as bacteria, whereas eukaryotes can be either single-celled, such as amoeba, or multicellular, such as some algae, plants, animals, and fungi. Eukaryotic cells contain organelles, including mitochondria, which provide energy for cell functions, chloroplasts, which create sugars by photosynthesis in plants, and ribosomes, which synthesize proteins. Cells were discovered by Robert Hooke in 1665, who named them after their resemblance to cells inhabited by Christian monks in a monastery. Cell theory, developed in 1839 by Matthias Jacob Schleiden and Theodor Schwann, states that all organisms are composed of one or more cells, that cells are. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Structure of a typical prokaryotic cell. The fundamental unit of structure and function in all living organisms, and that all cells come from pre-existing cells. Cell types. Main article, cell type. Cells are broadly categorized into two types: eukaryotic cells, which possess a nucleus, and prokaryotic cells, which lack a nucleus but have a nucleated region. Prokaryotes are single-celled organisms, whereas eukaryotes can be either single-celled or multicellular. Citation needed. Prokaryotic cells. Main article, prokaryote. Prokaryotes include bacteria and archaea, two of the three domains of life. Prokaryotic cells were the first form of life on Earth, characterized by having vital biological processes including cell signaling. They are simpler and smaller than eukaryotic cells, and lack a nucleus, and other membrane-bound organelles. The DNA of a prokaryotic cell consists of a single circular chromosome that is in direct contact with the cytoplasm. The nuclear region in the cytoplasm is called the nucleid. Most prokaryotes are the smallest of all organisms, ranging from 0.5 to 2.0 micrometers in diameter. 1. Page needed. A prokaryotic cell has 3 regions. Enclosing the cell is the cell envelope, generally consisting of a plasma membrane covered by a cell wall which, for some bacteria, may be further covered by a third layer called a capsule. Though most prokaryotes have both a cell membrane and a cell wall, there are exceptions such as mycoplasma, bacteria, and thermoplasma, archaea, which only possess the cell membrane layer. The envelope gives rigidity to the cell and separates the interior of the cell from its environment, serving as a protective filter. The cell wall consists of peptidoglycan in bacteria and acts as an additional barrier against exterior forces. It also prevents the cell from expanding and bursting, cytolysis, from osmotic pressure due to a hypotonic environment. Some eukaryotic cells, plant cells and fungal cells, also have a cell wall. Inside the cell is the cytoplasmic region that contains the genome, DNA, ribosomes and various sorts of inclusions. 2. The genetic material is freely found in the cytoplasm. Prokaryotes can explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Structure of a typical animal cell. Structure of a typical plant cell. Carry extrachromosomal DNA elements called plasmids, which are usually circular, linear. Bacterial plasmids have been identified in several species of spirochete bacteria, including members of the genus Borrelia, notably Borrelia burgdorferi, which causes Lyme disease. 3. Though not forming a nucleus, the DNA is condensed in a nucleid. Plasmids encode additional genes, such as antibiotic resistance genes. On the outside, some prokaryotes have flagella and pili that project from the cell's surface. These are structures made of proteins that facilitate movement and communication between cells. Eukaryotic cells. Main article, eukaryote. Plants, animals, fungi, slime molds, protozoa, and algae are all eukaryotic. These cells are about 15 times wider than a typical prokaryote and can be as much as 1,000 times greater in volume. The main distinguishing feature of eukaryotes as compared to prokaryotes is compartmentalization, the presence of membrane-bound organelles, compartments, in which specific activities take place. Most important among these is a cell nucleus, 2, an organelle that houses the cell's DNA. This nucleus gives the eukaryote its name, which means true kernel, nucleus. Some of the other differences are: the plasma membrane resembles that of prokaryotes in function, with minor differences in the setup. Cell walls may or may not be present. The eukaryotic DNA is organized in one or more linear molecules, called chromosomes, which are associated with histone proteins. All chromosomal DNA is stored in the cell nucleus, separated from the cytoplasm by a membrane. 2. Some eukaryotic organelles such as mitochondria also contain some DNA. Many eukaryotic cells are ciliated with primary cilia. Primary cilia play important roles in chemosensation, mechanosensation, and thermosensation. Each cilium may thus be viewed as a sensory cellular antennae that coordinates a large number of cellular signaling pathways, sometimes coupling the signaling to ciliary motility or alternatively to cell division and differentiation. 4. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Motile eukaryotes can move using motile cilia or flagella. Motile cells are absent in conifers and flowering plants. Citation needed: eukaryotic flagella are more complex than those of prokaryotes. 5. Comparison of features of prokaryotic and eukaryotic cells. Prokaryotes, eukaryotes. Typical organisms: bacteria, archaeoprotists, algae, fungi, plants, animals. Typical size: 1 m, 6, 10 100 m, 6. Type of nucleus: nucleated region, no. True nucleus: true nucleus with double membrane. DNA: circular, usually. Linear molecules: chromosomes, with histone. Proteins: RNA/protein. Synthesis: coupled in the cytoplasm. RNA synthesis in the nucleus. Protein synthesis in the cytoplasm. Ribosomes 50S and 30S, 60S and 40S. Cytoplasmic structure: very few structures. Highly structured by endomembranes and a cytoskeleton. Cell movement: flagella made of flagellin. Flagella and cilia containing microtubules. Lamellipodia and filopodia containing actin. Mitochondria: none, 1 to several thousand. Chloroplasts: none in algae and plants. Organization: usually single cells. Single cells, colonies, higher multicellular. Organisms with specialized cells. Cell division: binary fission, simple. Division: mitosis, fission or budding. Meiosis: chromosomes, single chromosome, more than one chromosome. Membrane: cell membrane and membrane-bound organelles. Many groups of eukaryotes are single-celled. Among the many-celled groups are animals and plants. The number of cells in these groups vary with species. It has been estimated that the human body contains around 37 trillion, 3.721013, cells, 7, and more recent studies put this number at around 30 trillion, 36 trillion cells in the male, 28 trillion in the female. 8. Subcellular components. All cells, whether prokaryotic or eukaryotic, have a membrane that envelops the cell, regulates what moves in and out, selectively permeable, and maintains the electric potential of the cell. Inside the Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Detailed diagram of lipid bilayer of cell. Membrane. Membrane: the cytoplasm takes up most of the cell's volume. Except red blood cells, which lack a cell nucleus and most organelles to accommodate maximum space for hemoglobin, all cells possess DNA, the hereditary material of genes, and RNA, containing the information necessary to build various proteins such as enzymes, the cell's primary machinery. There are also other kinds of biomolecules in cells. This article lists these primary cellular components, then briefly describes their function. Cell membrane. Main article: cell membrane. The cell membrane, or plasma membrane, is a selectively permeable, citation needed, biological membrane that surrounds the cytoplasm of a cell. In animals, the plasma membrane is the outer boundary of the cell, while in plants and prokaryotes, it is usually covered by a cell wall. This membrane serves to separate and protect a cell from its surrounding environment and is made mostly from a double layer of phospholipids, which are amphiphilic, partly hydrophobic and partly hydrophilic. Hence, the layer is called a phospholipid bilayer, or sometimes a fluid mosaic membrane. Embedded within this membrane is a macromolecular structure called the porosome, the universal secretory portal in cells, and a variety of protein molecules that act as channels and pumps that move different molecules into and out of the cell. 2. The membrane is semipermeable, and selectively permeable, in that it can either let a substance, molecule or ion, pass through freely, to a limited extent, or not at all. Citation needed, cell surface. Membranes also contain receptor proteins that allow cells to detect external signaling molecules, such as hormones. 9. Cytoskeleton. Main article, cytoskeleton. Further information, morphogenesis. The cytoskeleton acts to organize and maintain the cell's shape, anchors organelles in place, helps during endocytosis, the uptake of external materials by a cell, and cytokinesis, the separation of daughter cells after cell division, and moves parts of the cell in processes of growth and mobility. The eukaryotic cytoskeleton is composed of microtubules, intermediate filaments and microfilaments. In the cytoskeleton of a neuron, explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. A fluorescent image of an endothelial cell. Nuclei are stained blue, mitochondria are stained red, and microfilaments are stained green. Deoxyribonucleic acid, DNA. The intermediate filaments are known as neurofilaments. There are a great number of proteins associated with them, each controlling a cell's structure by directing, bundling, and aligning filaments. 2. The prokaryotic cytoskeleton is less well studied, but is involved in the maintenance of cell shape, polarity and cytokinesis. 10. The subunit protein of microfilaments is a small, monomeric protein called actin. The subunit of microtubules is a dimeric molecule called tubulin. Intermediate filaments are heteropolymers whose subunits vary among the cell types in different tissues. Some of the subunit proteins of intermediate filaments include vimentin, desmin, lamin, lamins A, B and C, keratin, multiple acidic and basic keratins, and neurofilament proteins, NFL, NFM. Genetic material. Main articles, DNA and RNA. Two different kinds of genetic material exist, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. Cells use DNA for their long-term information storage. The biological information contained in an organism is encoded in its DNA sequence. 2. RNA is used for information transport, for example, mRNA, and enzymatic functions, for example, ribosomal RNA. Transfer RNA. tRNA molecules are used to add amino acids during protein translation. Prokaryotic genetic material is organized in a simple circular bacterial chromosome in the nucleated region of the cytoplasm. Eukaryotic genetic material is divided into different, linear molecules called chromosomes inside a discrete nucleus, usually with additional genetic material in some organelles like mitochondria and chloroplasts. See endosymbiotic theory. A human cell has genetic material contained in the cell nucleus, the nuclear genome, and in the mitochondria, the mitochondrial genome. In humans, the nuclear genome is divided into 46 linear DNA molecules, called chromosomes, including 22 homologous chromosome pairs and a pair of sex chromosomes. The mitochondrial genome is a circular DNA molecule distinct from nuclear DNA. Although the mitochondrial DNA is very small compared to nuclear chromosomes, it codes for 13 proteins involved in mitochondrial energy production and specific tuners. Foreign genetic material, most commonly DNA, can also be artificially introduced into the cell by a process called transfection. This can be transient, if the DNA is not inserted into the cell's genome, or stable, if it is. Certain viruses also insert their genetic material into the genome. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Human cancer cells, specifically HeLa cells, with DNA stained blue. The central and rightmost cell are in interphase, so their DNA is diffuse and the entire nuclei are labeled. The cell on the left is going through mitosis and its chromosomes have condensed organelles. Main article, organelle. Organelles are parts of the cell that are adapted and / or specialized for carrying out one or more vital functions, analogous to the organs of the human body, such as the heart, lung, and kidney, with each organ performing a different function. 2. Both eukaryotic and prokaryotic cells have organelles, but prokaryotic organelles are generally simpler and are not membrane-bound. There are several types of organelles in a cell. Some, such as the nucleus and Golgi apparatus, are typically solitary, while others, such as mitochondria, chloroplasts, peroxismes and lysosomes, can be numerous, hundreds to thousands. The cytosol is the gelatinous fluid that fills the cell and surrounds the organelles. Eukaryotic cell nucleus, a cell's information center. The cell nucleus is the most conspicuous organelle found in a eukaryotic cell. It houses the cell's chromosomes, and is the place where almost all DNA replication and RNA synthesis, transcription, occur. The nucleus is spherical and separated from the cytoplasm by a double membrane called the nuclear envelope. Space between these two membranes is called perinuclear space. The nuclear envelope isolates and protects a cell's DNA from various molecules that could accidentally damage its structure or interfere with its processing. During processing, DNA is transcribed, or copied into a special RNA, called messenger RNA, mRNA. This mRNA is then transported out of the nucleus, where it is translated into a specific protein molecule. The nucleolus is a specialized region within the nucleus where ribosome subunits are assembled. In prokaryotes, DNA processing takes place in the cytoplasm. 2. Mitochondria and chloroplasts generate energy for the cell. Mitochondria are self-replicating, double membrane-bound organelles that occur in various numbers, shapes, and sizes in the cytoplasm of all eukaryotic cells. 2. Respiration occurs in the cell mitochondria, which generate the cell's energy by oxidative phosphorylation, using oxygen to release energy stored in cellular nutrients, typically pertaining to glucose, to generate ATP, aerobic respiration. Mitochondria multiply by binary fission, like prokaryotes. Chloroplasts can only be found in plants and algae, and they capture the sun's energy to make carbohydrates through photosynthesis. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Diagram of the endomembrane system. Endoplasmic reticulum. The endoplasmic reticulum, is a transport network for molecules, targeted for certain modifications and specific destinations, as compared to molecules that float freely in the cytoplasm. The has two forms, the rougher, which has ribosomes on its surface that secrete proteins into the and the smoother, which lacks ribosomes. 2. The smoother plays a role in calcium sequestration and release and also helps in synthesis of lipid. Golgi apparatus. The primary function of the Golgi apparatus is to process and package the macromolecules such as proteins and lipids that are synthesized by the cell. Lysosomes and peroxismes. Lysosomes contain digestive enzymes, acid hydrolases. They digest excess or worn-out organelles, food particles, and engulfed viruses or bacteria. Peroxismes have enzymes that rid the cell of toxic peroxides. Lysosomes are optimally active in an acidic environment. The cell could not house these destructive enzymes if they were not contained in a membrane-bound system. 2. Centrosome. The cytoskeleton organizer. The centrosome produces the microtubules of a cell, a key component of the cytoskeleton. It directs the transport through the and the Golgi apparatus. Centrosomes are composed of two centrioles which lie perpendicular to each other, in which each has an organization like a cartwheel, which separate during cell division and help in the formation of the mitotic spindle. A single centrosome is present in the animal cells. They are also found in some fungi and algae cells. Vacuoles. Vacuoles sequester waste products and in plant cells store water. They are often described as liquid-filled spaces and are surrounded by a membrane. Some cells, most notably amoeba, have contractile vacuoles, which can pump water out of the cell if there is too much water. The vacuoles of plant cells and fungal cells are usually larger than those of animal cells. Vacuoles of plant cells are surrounded by a membrane which transports ions against concentration gradients. Eukaryotic and prokaryotic. Ribosomes. The ribosome is a large complex of RNA and protein molecules. 2. They each consist of two subunits, and act as an assembly line where RNA from the nucleus is used to synthesize proteins from amino acids. Ribosomes can be found either floating freely or bound to a membrane, the rough endoplasmatic reticulum in eukaryotes, or the cell membrane in prokaryotes. 11. Plastids. Plastid are membrane-bound organelle generally found in plant cells and euglenoids, and contain specific pigments, thus affecting the color of the plant and organism. and these. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Pigments also helps in food storage and tapping of light energy. There are 3 types of plastids based upon the specific pigments. Chloroplasts contain chlorophyll and some carotenoid pigments which helps in the tapping of light energy during photosynthesis. Chromoplasts contain fat-soluble carotenoid pigments like orange carotene and yellow xanthophylls which helps in synthesis and storage. Leucoplasts are non-pigmented plastids and helps in storage of nutrients. 12. Structures outside the cell membrane. Many cells also have structures which exist wholly or partially outside the cell membrane. These structures are notable because they are not protected from the external environment by the cell membrane. In order to assemble these structures, their components must be carried across the cell membrane by export processes. Cell wall. Further information, cell wall. Many types of prokaryotic and eukaryotic cells have a cell wall. The cell wall acts to protect the cell mechanically and chemically from its environment, and is an additional layer of protection to the cell membrane. Different types of cell have cell walls made up of different materials. Plant cell walls are primarily made up of cellulose. Fungi cell walls are made up of chitin, and bacteria cell walls are made up of peptidoglycan. Prokaryotic capsule. A gelatinous capsule is present in some bacteria outside the cell membrane and cell wall. The capsule may be polysaccharide as in pneumococci, meningococci, or polypeptide as bacillus anthracis, or hyaluronic acid as in streptococci. Capsules are not marked by normal staining protocols and can be detected by India ink or methyl blue, which allows for higher contrast between the cells for observation. 1387. Flagella. Flagella are organelles for cellular mobility. The bacterial flagellum stretches from cytoplasm through the cell membrane, S, and extrudes through the cell wall. They are long and thick thread-like appendages, protein in nature. A different type of flagellum is found in archaea and a different type is found in eukaryotes. Fimbriae. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Prokaryotes divide by binary fission, while eukaryotes divide by mitosis or meiosis. A fimbria, plural fimbriae, also known as a pilus, plural pili, is a short, thin, hair-like filament found on the surface of bacteria. Fimbriae are formed of a protein called pilin, antigenic, and are responsible for the attachment of bacteria to specific receptors on human cells, cell adhesion. There are special types of pili involved in bacterial conjugation, cellular processes, replication. Main article, cell division. Cell division involves a single cell, called a mother cell, dividing into 2 daughter cells. This leads to growth in multicellular organisms, the growth of tissue, and to procreation, vegetative reproduction, in unicellular organisms. Prokaryotic cells divide by binary fission, while eukaryotic cells usually undergo a process of nuclear division, called mitosis, followed by division of the cell, called cytokinesis. A diploid cell may also undergo meiosis to produce haploid cells, usually 4. Haploid cells serve as gametes in multicellular Organisms, fusing to form new diploid cells. DNA replication, or the process of duplicating a cell's genome, 2, always happens when a cell divides through mitosis or binary fission. This occurs during the S phase of the cell cycle. In meiosis, the DNA is replicated only once, while the cell divides twice. DNA replication only occurs before meiosis. DNA replication does not occur when the cells divide the second time, in meiosis. 2.14. Replication, like all cellular activities, requires specialized proteins for carrying out the job. 2. DNA repair. Main article, DNA repair. Cells of all organisms contain enzyme systems that scan their DNA for damage and carry out repair processes when it is detected. Diverse repair processes have evolved in organisms ranging from bacteria to humans. The widespread prevalence of these repair processes indicates the importance of maintaining cellular DNA in an undamaged state in order to avoid cell death or errors of replication due to damage that could lead to mutation. E. coli bacteria are a well-studied example of a cellular organism with diverse well-defined DNA repair processes. These include nucleotide. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Excision repair, DNA mismatch repair, non-homologous end joining of double-strand breaks. Recombinational repair and light-dependent repair, photoreactivation. 15. Growth and metabolism. Main articles, cell growth, metabolism, and photosynthesis. Between successive cell divisions, cells grow through the functioning of cellular metabolism. Cell metabolism is the process by which individual cells process nutrient molecules. Metabolism has two distinct divisions: catabolism, in which the cell breaks down complex molecules to produce energy and reducing power, and anabolism, in which the cell uses energy and reducing power to construct complex molecules and perform other biological functions. Complex sugars can be broken down into simpler sugar molecules called monosaccharides, such as glucose. Once inside the cell, glucose is broken down to make adenosine triphosphate, ATP, 2. A molecule that possesses readily available energy, through two different pathways. In plant cells, chloroplasts create sugars by photosynthesis, using the energy of light to join molecules of water and carbon dioxide. Protein synthesis. Main article, protein biosynthesis. Cells are capable of synthesizing new proteins, which are essential for the modulation and maintenance of cellular activities. This process involves the formation of new protein molecules from amino acid building blocks based on information encoded in DNA/RNA. Protein synthesis generally consists of two major steps: transcription and translation. Transcription is the process where genetic information in DNA is used to produce a complementary RNA strand. This RNA strand is then processed to give messenger RNA, mRNA, which is free to migrate through the cell. mRNA molecules bind to protein RNA complexes called ribosomes located in the cytosol, where they are translated into polypeptide sequences. The ribosome mediates the formation of a polypeptide sequence based on the mRNA sequence. The mRNA sequence directly relates to the polypeptide sequence by binding to transfer RNA, tRNA, adapter molecules in binding pockets within the ribosome. The new polypeptide then folds into a functional three-dimensional protein molecule. Motility. Main article, motility. Unicellular organisms can move in order to find food or escape predators. Common mechanisms of motion include flagella and cilia. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Staining of a Caenorhabditis elegans highlights the nuclei of its cells. In multicellular organisms, cells can move during processes such as wound healing, the immune response and cancer metastasis. For example, in wound healing in animals, white blood cells move to the wound site to kill the microorganisms that cause infection. Cell motility involves many receptors, cross-linking, bundling, binding, adhesion, motor and other proteins. 16. The process is divided into 3 steps: protrusion of the leading edge of the cell, adhesion of the leading edge and de-adhesion at the cell body and rear, and cytoskeletal contraction to pull the cell forward. Each step is driven by physical forces generated by unique segments of the cytoskeleton. 17. 16. Navigation, control and communication. See also, cybernetics in biology. In August 2020, scientists described one-way cells, in particular cells of a slime mold and mouse. Pancreatic cancer-derived cells are able to navigate efficiently through a body and identify the best routes through complex mazes, generating gradients after breaking down diffused chemotractants which enable them to sense upcoming maze junctions before reaching them, including around corners. 18, 19, 20. Multicellularity. Main article, multicellular organism. Cell specialization / differentiation. Main article, cellular differentiation. Multicellular organisms are organisms that consist of more than one cell, in contrast to single-celled organisms. 21. In complex multicellular organisms, cells specialize into different cell types that are adapted to particular functions. In mammals, major cell types include skin cells, muscle cells, neurons, blood cells, fibroblasts, stem cells, and others. Cell types differ both in appearance and function, yet are genetically identical. Cells are able to be of the same genotype but of different cell type due to the differential expression of the genes they contain. Most distinct cell types arise from a single totipotent cell, called a zygote, that differentiates into hundreds of different cell types during the course of development. Differentiation of cells is driven by different environmental cues, such as cell-cell interaction, and intrinsic differences, such as those caused by the uneven distribution of molecules during division. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Stromatolites are left behind by cyanobacteria, also called blue-green algae. They are among the oldest fossils of life on Earth. This 1 billion-year-old fossil is from Glacier National Park in the United States. Origin of multicellularity. Multicellularity has evolved independently at least 25 times, 22, including in some prokaryotes, like cyanobacteria, myxobacteria, actinomycetes, or methanosarcina. However, complex multicellular Organisms evolved only in 6 eukaryotic groups: animals, fungi, brown algae, red algae, green algae, and plants. 23. It evolved repeatedly for plants, chloroplastida, once or twice for animals, once for brown algae, and perhaps several times for fungi, slime molds, and red algae. 24. Multicellularity may have evolved from colonies of interdependent organisms, from cellularization, or from organisms in symbiotic relationships. The first evidence of multicellularity is from cyanobacteria-like organisms that lived between 3 and 3.5 billion years ago. 22. Other early fossils of multicellular organisms include the contested Grippinea spiralis and the fossils of the black shales of the Paleoproterozoic Francevalian group. Fossil B formation in Gabon. 25. The evolution of multicellularity from unicellular ancestors has been replicated in the laboratory, in evolution experiments using predation as the selective pressure. 22. Origins. Main article: Evolutionary History of Life. The origin of cells has to do with the origin of life, which began the history of life on Earth. Origin of life. Further information: abiogenesis and evolution of cells. Small molecules needed for life may have been carried to Earth on meteorites, created at deep-sea vents, or synthesized by lightning in a reducing atmosphere. There is little experimental data defining what the first self-replicating forms were. RNA may have been the earliest self-replicating molecule, as it can both store genetic information and catalyze chemical reactions. 26. Cells emerged around 4 billion years ago. 27. 28. The first cells were most likely heterotrophs. The early cell membranes were probably simpler and more permeable than modern ones, with only a single fatty acid chain per lipid. Lipids spontaneously form bilayered vesicles in water, and could have preceded RNA. 29. 30. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. In the theory of semiogenesis, a merger of an archaean and an aerobic bacterium created the eukaryotes, with aerobic mitochondria, some 2.2 billion years ago. A second merger, 1.6 billion years ago, added chloroplasts, creating the green plants. 31. First eukaryotic cells. Main article: Eukaryogenesis. Eukaryotic cells were created some 2.2 billion years ago in a process called eukaryogenesis. This is widely agreed to have involved semiogenesis, in which archaea and bacteria came together to create the first eukaryotic common ancestor. This cell had a new level of complexity and capability, with a nucleus, 32, 33, and facultatively aerobic mitochondria. 31. It evolved some 2 billion years ago into a population of single-celled organisms that included the last eukaryotic common ancestor, gaining capabilities along the way, though the sequence of the steps involved has been disputed, and may not have started with semiogenesis. It featured at least one centriole and cilium, sex, meiosis and syngamy, paroxysms, and a dormant cyst with a cell wall of chitin and /or cellulose. 34, 35. In turn, the last eukaryotic common ancestor gave rise to the eukaryotes crown group, containing the ancestors of animals, fungi, plants, and a diverse range of single-celled organisms. 37. The plants were created around 1.6 billion years ago with a second episode of semiogenesis that added chloroplasts, derived from cyanobacteria. 31. History of research. Main article, Cell Theory. In 1665, Robert Hooke examined a thin slice of cork under his microscope, and saw a structure of small enclosures. He wrote, "I could exceeding plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of it were not regular." 38. To further support his theory, Matthias Schleiden and Theodor Schwann both also studied cells of both animal and plants. What they discovered were significant differences between the two types of cells. This put forth the idea that cells were not only fundamental to plants, but animals as well. 39. 1632–1723, Antony van Leeuwenhoek taught himself to make lenses, constructed basic optical microscopes and drew protozoa, such as Vorticella from rainwater, and bacteria from his own mouth. 40. 1665, Robert Hooke discovered cells in cork, then in living plant tissue using an early compound microscope. He coined the term cell, from Latin cellula, meaning small room. 41, in his book Micrographia, 1665. 42, 40. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Robert Hooke's Drawing of Cells in Cork. 1665. 1839, Theodor Schwann, 43, and Matthias Jacob Schleiden elucidated the principle that plants and animals are made of cells, concluding that cells are a common unit of structure and development, and thus founding the cell theory. 1855, Rudolf Virchow stated that new cells come from pre-existing cells by cell division, omnis cellula ex cellula. 1931, Ernst Ruska built the first transmission electron microscope, TEM, at the University of Berlin. 44, by 1935, he had built an M with twice the resolution of a light microscope, revealing previously unresolvable organelles. 1981, Lynn Margulies published Symbiosis in Cell Evolution, detailing how eukaryotic cells were created by semiogenesis. 45. See also cell cortex, cell culture, cellular model, cytonym, cytoresis, cytotoxicity, lipid raft, list of distinct cell types in the adult human body, outline of cell biology, parakaryon myogenesis, plasmolysis, syncytium, tunneling nanotube, volt, organelle. References: 1 Black, Jacqueline G. 2004. Microbiology. New York Chichester, Wiley. ISBN 978-0-471-42084-2. 2 ABCDEFGHIJKLMNOPQ. This article incorporates public domain material from What is a Cell? Science Primer. NCBI. 30 March 2004. Archived from the original on 8 December 2009. Retrieved 3 May 2013. 3 European Bioinformatics Institute, Karin's Genomes, Borrelia Burgdorferi, archived 6 May 2013 at the Wayback Machine, part of Toucan on the EBIEMBL database. Retrieved 5 August 2012. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 4 Sata, P. Christensen, Sren T. June 2008. Structure and function of mammalian cilia. Histochemistry and cell biology. 129, 6687693 do. 10.1007/S00418-008-04169. PMC 2386530. PMID 183652335. 1432-119X. 5 Blair, D.F. Dutcher, S.K. October 1992. Flagella in prokaryotes and lower eukaryotes. Current opinion in genetics and development. 2. 5756767 do. 10.1016/S0959-437X, 05, 80136-4. PMID 1458024. 6 A.B. Campbell Biology Concepts and Connections. Pearson Education. 2009 p. 320. 7 Biancani, Eva, Piovson, Alison, Fakin, Federica, Berardi, Alina, Casadia, Raffaella, Frey Betty. Flavia, Vital, Lorenza, Pellerai, Maria Chiara, Tassani, Simone, Piva, Francesco, Perezimodio. Soledad, 1 November 2013. An estimation of the number of cells in the human body. Annals of Human Biology. 40, 6463471 do. 10.301440/0301460.2013.807878 HDL 11585/1524451. ISSN 0301 4460. PMID 2382964. S2 SID 1624766. 8 Hatton, Ian A. Galbraith, Eric D. Merlio, Nuno SC. Meetinen, Timu P. Smith, Benjamin. MacDonald, Shanda, Jefferio. 26 September 2023. The Human Cell Count and Size Distribution. Proceedings of the National Academy of Sciences. 120, 39 E23030777120. Bibcode 2023 NAS 12003077H. Do 10.1073/NAS.23030777120. ISSN 0027-8424. PMC 105234666. PMID 377222043. 9 Gaitan, Arthur C. Hall, John E. 2016. Gaitan and Hall Textbook of Medical Physiology. Philadelphia, Elsevier Saunders. pp. 93937. ISBN 978-1-4557-7005-2. OCLC 1027900365. 10 carat Michi, Kaya. Low, J. 2006. Dynamic filaments of the bacterial cytoskeleton. Annual Review of Biochemistry. 75, 467492. Do. 10.1001.biochem.75.1030004.14245. PMID 167564999. S2 SID 4550126. 11 carat Menitrit, Jean-François, Skaletsky, Julia, Clements, William M. It al. December 2007. Ribosome. Binding of a single copy of the Sec-Y complex, Implications for Protein Translocation, PDF. Molecular. Cell. 28, 6 1083 1092 do. 10.1016/j.molchel.2007.10.034. PMID 181589004. Archived, PDF. From the original on 21 January 2021. Retrieved 1 September 2020. 12 carat Sato, N. 2006. Origin and Evolution of Plastids, Genomic View on the Unification and Diversity of Plastids. In Wise, R. Huber, J.K. eds. The Structure and Function of Plastids. Advances in Photosynthesis and Respiration. Volume 23. Springer. pp. 75102. Do. 10.101781402040610610_4. ISBN 978-1-4020-4060-3. 13 carat Prokaryotes. Nunes. 1996. ISBN 978-0080984735. Archived from the original on 14 April 2021. Retrieved 9 November 2020. 14 carat Campbell Biology Concepts and Connections. Pearson Education. 2009 p. 138. 15 carat Snestad, D. Peter, Simmons, Michael J. Principles of Genetics, 5th ed. DNA Repair Mechanisms, pp. 364368. 16 carat A. B. Ananthakrishnan, R. Ulika, A. June 2007. The Forces Behind Cell Movement. International Journal of Biological Sciences. 3, 5. Biolsh.org, 303317. Do. 10.7150/IJPS.3.303. PMC 1893118. PMID 175896565. 17 carat Alberts, Bruce, 2002. Molecular Biology of the Cell, 4th ed. Garland Science. pp. 973975. ISBN 0815340729. 18 carat Willingham, Emily. Cells solve an English hedge maze with the same skills they use to traverse the body. Scientific American. Archived from the original on 4 September 2020. Retrieved 7 September 2020. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 19. How cells can find their way through the human body. phys.org. Archived from the original on 3 September 2020. Retrieved 7 September 2020. 20. Tweedy, Luke, Thomason, Peter A. Pask, Peggy I. Martin, Kirsty, Mashiski, Laura M. Zagnani. Michelle, Insel, Robert H. August 2020. Seeing around corners, cells solve mazes and respond at a distance using attractant breakdown. Science. 369, 6507 9792. Do. 10.1126 /science.aa9792. PMID 328553111. S2 SID 2213425551. Archived from the original. On the 12th of September 2020. Retrieved the 13th of September 2020. 21. Becker, Wayne M. It al. 2009. The World of the Cell. Pearson Benjamin Cummings. p. 480. ISBN 978-0321554185. 22. ABC Grossberg, R.K. Strathman, R. 2007. The Evolution of Multicellularity, a Minor Major. Transition. PDF. A new Revicolivol SYST. 38, 621654. Do. 10.1146.36.102403.114735. Archived from the original, PDF, on 4 March 2016. Retrieved 23 December 2013. 23. Popper, Zoe. Michel, Gervin, Herve, Cecile, et al. 2011. Evolution and Diversity of Plant Cell. Walls, From Algae to Flowering Plants, PDF. Annual Review of Plant Biology. 62, 567590. Do. 10.1146/ANAVAR plant 042110-103809. HDL 10379/6762. PMID 21351878. S2 SID 11961888. Archived, PDF, from the original on 29 July 2016. Retrieved 23 December 2013. 24. Borner, John Tyler, 1998. The Origins of Multicellularity, PDF. Integrative Biology. 1, 12736. Do. 10.1002/CISI 1520-6602, 1998, 113.0.co, 2-6. ISSN 1093-4391. Archived. From the original, PDF, 0.2 MB, on 8 March 2012. 25. Albani, Abdurazak L., Benson, Stefan, Camfield, Donald E. It al. July 2010. Large Colonial. Organisms with coordinated growth in oxygenated environments 2.1 GYR. Nature. 466, 7302 100. 104 Bibcode, 2010 Nature.466 100 Do. 10.1038 /Nature 09166. PMID 20596019. S2 SID 4331375. 26 Orgel, L.E. December 1998. The Origin of Life, a Review of Facts and Speculations. Trends in Biochemical Sciences. 23, 12491495 do. 10.1016 /S0968-0004, 98, 01300-0. PMID 9868373. 27 Dodd, Matthew S. Pepino, Dominic, Gren, Tor, et al. 1 March 2017. Evidence for early life in Earth's oldest hydrothermal vent precipitates. Nature. 543, 7643 6064. Bibcode, 2017 Nature.543, 60d. Do. 10.1038 Nature 21377. PMID 28252057. Archived from the original on 8 September 2017. Retrieved 2 March 2017. 28 Betts, Holly C. Puttick, Mark N. Clark, James W. Williams, Tom. Donahue, Philip C. J. Pisani. Davide, 20 August 2018. Integrated genomic and fossil evidence illuminates life's early evolution and eukaryote origin. Nature, Ecology and Evolution. 2, 10 1556 1562. Bibcode, 2018 NATIE, 2.1556b. Do. 10.1038 /S41559-018-0644X. PMC 6152910. PMID 301275539. 29 Griffiths, G. December 2007. Cell Evolution and the Problem of Membrane Topology. Nature. Reviews. Molecular Cell Biology. 8, 12 1018 1024 do. 10.1038 /NRM 2287. PMID 17971839. S2 SID 31072778. 30 First cells may have emerged because building blocks of proteins stabilized membranes. Science Daily. Archived from the original on 18 September 2021. Retrieved 18 September 2021. 31 ABC Lator, A. Durban, A. Moya, A. Perito, J. 2011. The Role of Symbiosis in Eukaryotic Evolution. In Gargaud, Muriel, Lopez Garcia, Purification, Martin, H. eds. Origins and Evolution of Life, an Astrobiological Perspective. Cambridge, Cambridge University Press. pp. 326339. ISBN 978 0-521-76131-4. Archived from the original on the 24th of March 2019. Retrieved the 27th of August 2017. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 32 McGrath, Casey, the 31st of May 2022. Highlight: Unraveling the Origins of Luca and Luca on the Tree of Life. Genome Biology and Evolution. 14, 6 evac 072 do 10.1093/GBE/evac 072. PMC 9168435. 33 Weiss, Madeline C. Souza, FL. Mjavik, N. It al. 2016. The Physiology and Habitat of the Last Universal Common Ancestor, PDF. Nature Microbiology. 1, 916116. Do. 10.1038/microbiol.2016.116. PMID 27562259. S2 SID 2997255. 34 Lender, B.S. May 2020. Predatory Protists. Current Biology. 30, 10R5 Do. 10.1016/j.cub.2020.03.052. PMID 324284991. S2 SID 218710816. 35 Strassert, Georgian F.H. Irisary, Iker, Williams, Thomas. Berkey, Fabian, the 25th of March 2021. A molecular timescale for eukaryote evolution with implications for the origin of red algal-derived plastids. Nature. Communications. 12, 1/1879. Bibcode 2021 Natco 12.1879s. Do. 10.1038/S41467-021-22044Z. PMC 7994803. PMID 337679194. 36 Gabalden, T. October 2021. Origin and Early Evolution of the Eukaryotic Cell. Annual Review of Microbiology. 75, 1631647 do. 10.1146/An of Micro 090817-062213. PMID 343433017. S2 SID 236916203. 37 Wois, C.R., Kandler, Otto, Wheelies, Mark L. June 1990. Towards a Natural System of Organisms. Proposal for the domains Archaea, Bacteria, and Eukarya. Proceedings of the National Academy of Sciences of the United States of America. 87, 12 4576 4579. Bibcode 1919as 87.4576W. Do. 10.1073/NAS.87.12.4576. PMC 54159. PMID 2112744. 38 Hook, Robert, 1665. Observation 18. Micrographia. 39 Matten, Anthea, 1997. Cells Building Blocks of Life. New Jersey, Prentice Hall. pp. 44-45. The Cell Theory. ISBN 978-0134234762. 40 A.B. Jest, H. 2004. The Discovery of Microorganisms by Robert Hooke and Antony van Leeuwenhoek, Fellows of the Royal Society. Notes and Records of the Royal Society of London. 58, 2. 187201. Do. 10.1098/RSNR.2004.0055. PMID 15209075. S2 SID 8297229. 41 The Origins of the Word Cell. National Public Radio. 17 September 2010. Archived. From the original on 5 August 2021. Retrieved 5 August 2021. Celler. A Latin Dictionary. Charlton T. Lewis and Charles Short. 1879. ISBN 978-1999855789. Archived from the original on 7 August 2021. Retrieved 5 August 2021. 42 Hooke, Robert, 1665. Micrographia. London, Royal Society of London. p. 113. I could exceedingly plainly perceive it to be all perforated and porous, much like a honeycomb, but that the pores of it were not regular. These pores, or cells, were indeed the first microscopical pores I ever saw, and perhaps, that were ever seen, for I had not met with any writer or person, that had made any mention of them before this. Hooke describing his observations on a thin slice of cork. See also, Robert Hooke, archived 6 June 1997 at the Wayback Machine. 43 Schwann, Theodore, 1839. Mikroskopisch Untersuchungen über die Übereinstimmung in der Struktur und dem Wachstum der Tiere und Pflanzen. Berlin, Sander. 44 Ernst Rusker, January 1980. The Early Development of Electron Lenses and Electron Microscopy. Applied Optics. Volume 25. Translated by T. Mulvey. p. 820. Bibcode, 1986 APOPT 25820R. Do. 10.1364/AO.25.000820. ISBN 978-3-7776-036-3. 45 Cornish Bowden, Athol, 7 December 2017. Lynn Margulies and the Origin of the Eukaryotes. Journal of Theoretical Biology. The Origin of Mitosing Cells, 50th Anniversary of a Classic Paper by Lynn Sargan, Margulies. 434, 1. Bibcode, 2017 JTB.434, 1c. Do. 10.1016/J.B.2017.09.027. PMID 28992902. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Further reading. Alberts, Bruce, Johnson, Alexander, Lewis, Julian, Morgan, David, Raff, Martin, Roberts, Keith, Walter. Peter, 2015. Molecular Biology of the Cell, 6th ed. Garland Science. p. 2. ISBN 978-0815344322. Alberts, B. It al. 2014. Molecular Biology of the Cell, 6th ed. Garland. ISBN 978-0815344322. Archived from the original on 14 July 2014. Retrieved 2016-07-06. The 4th edition is freely available. Archived the 11th of October 2009 at the Wayback Machine from National Center for Biotechnology Information. Bookshelf. Lodish, Harvey, et al. 2004. Molecular Cell Biology, 5th ed. New York, W. H. Freeman. ISBN 978. 0716743668. Cooper, G. M. 2000. The Cell, A Molecular Approach, 2nd ed. Washington, D.C., ASM Press. ISBN 978-0878931026. Archived from the original on 30 June 2009. Retrieved 30 August 2017. External links. MB Info descriptions on cellular functions and processes. Inside the Cell, archived 20 July 2017 at the Wayback Machine, a science education booklet. By National Institutes of Health, in PDF and EPUB. Cell Biology in the Biology Project of University of Arizona. Center of the Cell Online. The Image and Video Library of the American Society for Cell Biology, archived 10 June 2011 at The Wayback Machine, a collection of peer-reviewed still images, video clips and digital books that illustrate the structure, function and biology of the cell. Wormweb.org, interactive visualization of the C. elegans cell lineage. Visualize the entire cell lineage tree of the nematode C. elegans. Categories: Cell Biology, Cell Anatomy 1665 in Science. Explore our developer-friendly HTML to PDF API printed using PDF Crowd. HTML to PDF.